Regulation of gene transcription by hormones. Hormones, the chemical messengers of the body, play a pivotal role in regulating gene transcription, the process by which DNA is converted into RNA, which ultimately leads to protein synthesis. This intricate interplay between hormones and gene expression is essential for various physiological processes, including growth, development, metabolism, and reproduction. Mechanisms of hormonal regulation. The mechanisms by which hormones regulate gene transcription vary depending on the type of hormone. Steroid hormones. Intracellular receptors. Steroid hormones, such as estrogen, testosterone, and cortisol, are lipid-soluble and can easily diffuse through the cell membrane. Inside the cell, they bind to specific intracellular receptors. DNA binding. The hormone receptor complex acts as a transcription factor, binding to specific DNA sequences called hormone response elements, HREs, located near the target genes. Gene activation or repression. Binding of the hormone receptor complex can either activate or repress gene transcription, depending on the specific hormone and target gene. Peptide and protein hormones. Membrane receptors. Peptide and protein hormones, like insulin and growth hormone, are water-soluble and cannot cross the cell membrane directly. Instead, they bind to specific receptors on the cell surface. Signal transduction. Binding of the hormone to its receptor triggers a cascade of intracellular signaling events, often involving second messengers like cyclic AMP, CAMP, or calcium ions. Transcription factor activation. These signaling pathways ultimately lead to the activation or inhibition of transcription factors, which then bind to DNA and regulate gene expression. Factors affecting hormonal regulation. Hormone concentration. The concentration of a hormone in the bloodstream influences its ability to bind to receptors and activate gene transcription. Receptor sensitivity. The sensitivity of hormone receptors can vary depending on factors like tissue type, developmental stage, and exposure to other hormones. Co-regulators. Many proteins, called co-activators and co-repressors, interact with hormone receptor chromatin structure. The accessibility of DNA to transcription factors is influenced by chromatin structure, which can be modified by various factors, including histone modifications and DNA methylation. Examples of hormonal regulation: growth and development. Growth hormone stimulates the production of growth factors, leading to bone and muscle growth. Metabolism. Insulin regulates glucose uptake and metabolism by controlling the expression of genes involved in glucose transport and glycogen synthesis. Reproduction. Estrogen and progesterone regulate the menstrual cycle and pregnancy by controlling the expression of genes involved in endometrial growth, ovulation, and implantation. Implications of hormonal regulation. Dysregulation of hormone-mediated gene transcription can lead to various diseases, including diabetes, cancer, and reproductive disorders. Understanding the molecular mechanisms underlying hormonal regulation is crucial for developing new therapeutic strategies for these conditions.